Hello, and thank you for joining our virtual presentation today for the 5 days of stem cells. My name is Matt Poling. I'm a product manager for genome editing with Thermo Fisher Scientific. I joined Thermo Fisher in 2015 and currently support the CRISPR-Cas9 and Talon genome editing tool portfolio. Today, I'm going to be presenting advanced tools for knock-in genome editing in iPSC cells. Before I begin, I did want to take an opportunity and recognize all of the scientists that I work with in our gene editing, R&D, and product development groups. I currently support a team of 29 full-time genome editing scientists. This is a really dedicated group out of our Carlsbad, California site that's really focused on advancing these tools and the application of genome editing for life scientists. They spend a lot of time looking to further refine and optimize these tools and make them accessible to a wide range of scientists that use Thermo Fisher reagents. They focus on functional genomics, especially trying to understand various gene pathways and developing tools for that, with a specific focus in stem cell biology and stem cell engineering, which we'll talk about in this presentation as well. Our Carlsbad, California site is one of our major North America R&D hubs. We work closely with the delivery and transfection teams, as well as have access to our ion torrent next generation sequencing team. And I'll be showing you some data from that group as well. For our presentation today, we'll go over some 5 main agenda items. The first is we'll do a very quick review of genome editing. We will then talk briefly about CRISPR-Cas9 and specifically our wild type and our high fidelity Cas9 that's currently under development. We'll go through and I'll demonstrate our TrueDesign genome editor platform. This is a design software that can assist life scientists with designing genome editing experiments and getting all the required materials for those experiments to be conducted in the lab. I'll show you some application data with our stem cells and what we've been able to do before, and kind of an early look at where we're going with this technology in stem cells. And then lastly, we'll kind of finish our presentation about talking about moving into the clinic and beyond with some programs that we have looking to get products in our CTS portfolio of reagents. So to start off, I'm going to do a review of genome editing. And what we have on the screen here is a cartoon representation of what's occurring at the molecular level when a genome edit is occurring. What we have is a double-stranded piece of DNA there, and that gray circle is representing an engineered nuclease that's going to come in and cleave that DNA, causing a double-stranded break. When that double-stranded break occurs, the cell is going to attempt to repair it, and when it does, there's generally 2 main pathways that are involved. The first is the non-homologous end-joining pathway, or NHEJ, that will cause gene knockouts. These can cause premature stop codons by frame shifts, and these are generally what are used to make knockout models or do genomic screening. The second is going to be the homology-directed repair, or HDR pathway, and this is what's done to do gene insertions or general knock-in editing. For HDR to occur, we require the presence of a donor molecule that's going to have the new DNA that needs to be added to the genome, with then left and right homology arms that are going to be the same sequence flanking that break site that allows that donor template to be incorporated in the genome at that break site. This is done to create reporter genes, make fusion proteins, disrupt functional domains, and cause specifically SNP changes in disease models, which is a particular focus of stem cell biologists. For these pathways to be successfully reproduced in the lab, there's generally 3 main materials that are required for this. The first is going to be a specific and efficient nuclease system. The second is going to be a repair template with that desired change. And third is going to be a method to deliver them into the cells. Ultimately, we need to get this entire payload, that being the nuclease system and the repair template, into the cells, and kind of the collection of these materials together is what's needed to successfully get this material into the cells and cause that edit to occur at the molecular level. One of the most popular and arguably the most famous of these engineered nuclease systems is going to be the CRISPR-Cas9 system. This is an RNA-guided DNA nuclease system. This was originally identified as part of the bacterial adaptive immune response pathway, a way for bacteria to combat phage infections and go ahead and cleave that DNA. And what scientists have done is they have understand this molecular pathway and have co-opted it to be used for eukaryotic engineering. Ultimately, what's needed for this is we have an RNA-guided DNA nuclease system. The nuclease activity is conducted by the Cas9 protein that has the endonuclease domain, and the targeting of that is done by the guide RNA sequences, that CRISPR RNA sequence, and the tracer RNA molecules that act as a duplex or as a single guide RNA when done synthetically and introduced into cells. The design for this is dependent on a protostacer in the Streptococcus pyogenes system is going to be that NGG, and this determines where CRISPR-Cas9 cut. We're ultimately looking for a 20 nucleotide sequence that's going to be upstream of that NGG motif, and that dictates where Cas9 can bind and ultimately cleave the DNA. And when this cleavage occurs, it causes blunted DNA breaks that then get repaired either by blunt ligation, which can sometimes lead to insertions and deletions that lead to the endels and those frameshift mutations, or when a donor is present, there can be some chewback and donors can be incorporated into the genome by homology-directed repair. And for scientists to use this in the lab, they need to be introducing that Cas9 and guide RNA molecules into their cells. And this can be done by a plasmid format, as RNA molecules and mRNA Cas9 and a synthetic guide RNA, or what we favored is those RNP formats, a ribonucleoprotein format, where we have a recombinant Cas9 protein and a synthetic guide RNA. And to that end, I want to take an opportunity to discuss our TrueCut Cas9 protein. This is a protein that we had engineered and developed in 2016 and 2017. And that has been available to scientists since then, that delivers consistently high editing efficiency. We manufacture this at a really high quality under ISO 13485 standards. We'll talk about those quality standards a little bit later in the presentation when we talk about moving to the clinic. And we have a number of validated protocols and demonstrations of significant improvement in the on-target activity. So when we look at these charts here, we can see that at multiple loci, at multiple cell types, we can show very robust genome editing activity, very high rate of forming indels, those insertions and deletions that are indicative of genome editing occurring at multiple loci in multiple cell types. More of this data is available on thermofisher.com if you're looking to see more, and we have a number of application notes to this effect. But what we really wanted to do after we had developed the TrueCut Cas9 protein was understand how we can make a better Cas9. What can we do to further improve the performance of this enzyme and really give it the specificity and activity that's needed for successful genome editing? If we look at this chart here and we plot activity on the y-axis and specificity on the x-axis, our wild-type Cas9 is at the peak of that activity. You know, we're normalizing it here, and we can see that, you know, we've got the most activity for the wild-type Cas9. And as we look at, you know, protein clones that have more specificity, we see that activity drops off. The more specific a Cas9 becomes due to introducing various point mutations, that activity will drop off. And this is a figure that's reproduced from a publication that came out earlier this year. And we've seen this in our hands as well, that as you increase that specificity, that activity drops off. But the goal that we sought out in product development here was to try to find a Cas9 that has that increased specificity without removing or reducing any of that on-target activity. A lot of times the way that these specificity characteristics are built is by doing point mutations to the active site, and that can ultimately reduce the overall catalytic activity of the enzyme. But how can we find a way to find the highest activity with the highest specificity? And so what the team did is we looked at a number of characterized point mutations of the Cas9 enzyme, and then we screened that and challenged it by NGS to try to find the best enzyme for off-target identification through next-generation sequencing. For this, we're using a modified version of the guide-seq technique that we call TEG-seq. And what we're showing here is kind of an output from those guide-seq experiments. Ultimately, what we're looking at here is we have an on-target site, that's the one that's highlighted in green, and all of the various off-targets that have occurred that we can identify by next-generation sequencing. When we look at our wild-type Cas9, we can see, again, really nice cleavage at that on-target site, but all of this off-target activity occurring across the genome. And when we count this out, we're looking at about 34 off-target sites occurring with this guide RNA. So if you're using Cas9, a wild-type Cas9, and this guide RNA, you're going to have about 34 off-targets that need to be contended with in this cell line. But when we look at other high-fidelity enzymes, such as the competitor I enzyme here, we can see that actually it's able to successfully eliminate about half of those off-target events. We're reducing that down to about 14 off-target events. So we're getting a much cleaner profile with this competitor I enzyme. But what we were able to identify was a novel clone that we're calling our Invitrogen HiFi, where we've actually eliminated over 75% of those off-target events for this HEC4 loci, where I've got kind of zeros across the board here for the Invitrogen HiFi, with only a few off-target sites remaining for this particular guide RNA, which is known to have a pretty poor off-target profile. Again, showing a significant reduction of those off-target events. And this is just one example. We've done a really wide array of this across numerous loci in several different cell types. And when we kind of normalize this out, what we're able to show here is that we're consistently able to eliminate greater than 50% of those off-target events. Again, looking at the wild-type enzyme, normalizing that to 100%, the competitor I enzyme reducing about 30% of those off-target events, again, showing improvement, but not as much as what we're able to see with the Invitrogen HiFi enzyme, where we're able to eliminate greater than 50%, almost 60% of those off-target events kind of across the board. And what I talked about a couple slides ago was that when you reduce these off-target events, when you increase the specificity, you sometimes sacrifice on-target cutting. And actually, we're able to show that we're able to maintain about 80% of the enzyme's activity, the on-target cleavage activity, also as identified by next-generation sequencing at these on-target loci that are matched with the off-targets that we're looking at here. So what we've found here, and what we're continuing to develop and plan on making commercially available next year, is a novel HiFi enzyme that has, the most preserved on-target cutting while eliminating the majority of those off-target events. And what we have for this year is we actually have this protein available now through an early access program. Again, we're showing that significant reduction in off-target events, and we're maintaining that maximum on-target cleavage. We have this available in 3 pack sizes now, and this can be ordered through your cell biology specialist or through our, you can email us at gemservices@thermofisher.com. Again, so this is our new Cas9 protein that we're currently bringing into production and will have available globally next year. And if you're interested in early access to this program, please contact us at gemservices@thermofisher.com. Switching gears a little bit to talk about kind of the next product that's needed, or material that's needed for successful knock-in editing, is going to be the donor template. Again, when we're doing homology-directed repair and we've got cleavage of the DNA, if we want to be introducing new DNA, we have to provide that to the cells at the same time that the editings occur so that it can be incorporated into the genome. And there's really 4 main types of donor templates that are used by scientists today. The first is single-stranded DNA. This is generally used for small insertions or small changes. Generally have to be less than 200 nucleotides long to be synthesized. And this is really what's ideal and what we recommend for any small changes, SNP changes, doing amino acid substitutions, etc. Can be done with single-stranded DNA. The second is double-stranded DNA, linear double-stranded DNA. This can be used for larger insertions, generally in the 200 to 5 kb range, the 200 base pair to 5,000 base pair range. These can be linear plasmid fragments or produced by PCR. And these are what will be used for gene insertions, fusions, adding reporter genes, anything that requires, a larger amount of DNA to be incorporated into the genome. The next one is plasmid DNA. This can be used and is used by a lot of investigators for very large insertions. Up to 10 kb, we've seen reports of knock-in with that. And that's because, when you're trying to do these larger knock-ins, you obviously need to have a much larger template. And plasmid DNA can be effectively delivered in a circularized form and then incorporated into the genome. Again, these are generally manufactured by doing some sort of DNA synthesis or subcloning into shuttle vectors. And then the last one is going to be viral vectors. Specifically, AAV has become very popular for genome editing. This does have a payload limit of about 4.5 kb and does require a lengthy and tricky viral production process. So this is generally reserved for more difficult to transfect cells that require transduction. And there is generally a trade-off an investigator has to make in terms of how big of a payload they want to provide, as that is capped at around the 4 kb mark. Our work generally focuses on that single-stranded and double-stranded donor templates, and the data I'm going to be showing throughout the rest of the presentation is focusing on those two donor types. So when we think about single-stranded and double-stranded DNA donors, generally there's a lot of open questions that investigators will have about how to use this particular material. How close to the PAM site does my insertion need to be? You know, how long do my homology arms need to be? Should I be using an asymmetric donor? Should I be using single-stranded versus double-stranded? And ultimately, kind of the unifying question here is, can you help me design my donor? What does my donor need to look like so that I can have the most effective knock-in? And so what our R&D group was able to do was really simplify this and provide universal rules for donor design. We established consistent and simple rules for HDR donors, and then to communicate this and have this available, we developed a novel design tool with an intuitive user interface to make it easy for investigators to pick up and start doing their own donor design. And we made this tool free and available to all scientists by putting it on thermofisher.com in our Connect platform, which is our cloud. And the R&D data that kind of went into all of this design, we published in the Journal of Biotechnology. And if you're interested in reading more about the nitty-gritty R&D data, I encourage you to go look that up. It's available on thermofisher.com/TrueDesign. But ultimately, our design rules can be simplified down into three basic rules. The first is make sure that your DNA break, that double-stranded break, is less than 10 bases from the desired edit site. Wherever you plan on editing the genome, wherever you plan on inserting or changing the DNA, you want to make sure that you're cutting less than 10 bases from that site is preferred to get that highest efficiency. Cutting, significantly far away does not favor a successful repair. So we really want to make sure that we're restricted to that 10 base pair region. The second is to use the most efficient enzyme possible. You want to be using the best CRISPR-Cas9 guide RNA system or using a TALEN effector nuclease system to give the most efficient cleavage. And then lastly, use at minimum 30 nucleotide homology arms. There's a lot of data in the literature, suggesting various homology arm lengths, from 30 up to, several kb. And what we found is that actually those 30 nucleotide homology arms is really what's sufficient to get a successful knock-in, and that adding more doesn't necessarily increase the editing efficiency for these knock-ins, and those templates can be much more difficult to produce than a 30 nucleotide one. So generally we recommend at minimum 30 nucleotide homology arms, and you don't actually need much more than that to get a successful knock-in. And so to that, we've put these rules into the back-end engine for our TrueDesign Genome Editor software. This is a platform that we have available on thermofisher.com that allows users to create accurate and more successful knock-in experiments. What we allow users to do is to search for their particular gene of interest. We have the entire human genome loaded up, and you can find your particular gene and all of the associated transcripts for that to edit. You go ahead and craft your edit. We display that intron and exonic regions as well as the amino acids, and I'll show some more close-up screenshots of that in the future slides. You can go in and make specific edits, change amino acids, do insertions, deletions, make SNP changes, all within that tool. And then lastly, once you put in all that information for us, what kind of specific change you want to make, the tool will go ahead and complete the design. It'll give you the most recommended guide RNA sequence to cut at that site, as well as design a corresponding donor that'll make that edit for you. So looking more at the tool here, what we do is we ask you to provide a gene symbol or specific chromosomal coordinates that you're looking to edit. We'll then display all of the transcripts that are related to that particular locus. For this example here, I'm editing the REL-A gene, one of the subunits of NF-kappa B. And we can see that there's 4 variants of the REL-A gene here. When we select that first variant and we bring that up, we now see a topology of the REL-A gene across the genome. We can see the exons, we can see the introns, we can distinguish between protein coding regions and non-protein coding regions. And for this example, we're focused in on that C-terminus of the REL-A gene right before that stop codon. And what we're going to be doing here is doing an insertion of a 24 base pair sequence. And when we do that 24 base pair sequence, we can see the tool revising refreshing and showing the insertion of that DNA into the genome that would be accomplished with these particular tools. So again, here we're doing a 24 base pair insertion. This is actually the flag epitope, that DYK, DDDK sequence that's frequently used to make flag tag fusion proteins is the example that we're doing here. We have that flag sequence inserted. And then we're going to go ahead and complete the design. The tool will identify that that's your proposed insertion site. It'll identify and recommend guide RNAs based on that locus and where we want to be, specifically cutting the DNA. And then it'll ultimately give you a donor recommendation, a donor molecule that has those minimal left and right homology arms that we talked about a couple slides ago, with that, flag tag sequence being, in the center of the donor there. You can see what we've annotated. We've got those left and right homology arms and that DYK, DDDK sequence occurring right in the middle of the donor. And then what you can do from the tool is you can export all of this and save it for your digital lab notebook, or we also have the opportunity to add all of these donors, the Cas9, the guide RNA, everything that's needed to successfully complete this, add that to a Thermo Fisher doc cart for you or your lab manager to purchase and then use in your lab. So talking more about stem cells and getting kind of to the theme of 5 days of stem cells, I wanted to also show an example of how we can use this tool to create specific stem cell type changes. For this particular example, and for what a lot of stem cell researchers are doing, we're looking at making stem cells from either healthy donors or patient donors and looking at specific disease SNP changes, either correcting those SNP changes or introducing mutations into healthy donor cells to try to reproduce disease phenotypes. And we've published a couple different times now a variety of SNP changes that are related to disease models and our various indel rates and homology-directed repair rate for those. One of the more popular ones is the LRRK2 G2019S SNP change, and I'm going to show you how you can reproduce the design that we demonstrated here using TrueDesign Genome Editor. So same idea as before. If we're looking to do a mutation in the LRRK2 gene, doing that G2019S SNP change, we're going to go and search for that gene. We're able to pull up all the transcripts related to LRRK2. We can go in, identify that specific amino acid, that glycine, and we're going to do a SNP change here to change that G to an A. We're now got that glycine changing into a serine, so that's that G2019S annotation. And then once we've inputted that, the tool will go ahead and give us the appropriate guide RNAs. We see that there's one guide RNA that's recommended for this locus. And we can see that the donor that comes out of the tool has that SNP change in it. We've got that glycine to serine point mutation, that SNP change. And therefore, this donor used with this guide RNA, when delivered into your stem cells, would be able to successfully convert that and produce this SNP change in what would be considered a healthy donor iPSC line. So quick summary for this, our TrueDesign Genome Editor is a great free online tool that allows investigators to search for their gene, go ahead and craft the particular edit that they're looking to introduce in their cells, and then ultimately the tool will complete the design, giving them the appropriate guide RNA sequence as well as a matching donor sequence to accomplish this in their cells in the lab. Now, when we talk about editing in stem cells, kind of our third main consideration here is going to be that the delivery and having successful, delivery of the payload for genome editing into the stem cells. And generally what we recommend for that is using an electroporation system such as the Neon. We need a way to effectively deliver the Cas9, the guide RNA, the donor molecule into these cells at a high efficiency so that we can find successful clones. And to that end, we do recommend the Neon. An alternative path to that is using something like our Cas9 iPSC line. We have an iPSC line that has been transduced with a virus to express for Cas9, and now this gives a high expression of Cas9 at all times and makes the delivery of the payload a little bit easier because we can just deliver the guide RNA and the donor molecule with the Neon system or by liposuction using a lipofectamine reagent. But for either of these, ultimately, we want to make sure that we're using a system that's going to be able to effectively deliver that payload into the cells. Delivering Cas9 in a way that's ineffective will lead to a low editing efficiency and make a lot of work more difficult, make it a lot of difficult work for the user. If you're looking to get started in editing your stem cells, we actually have a really nice kind of starter package here, being the Pluripotent Stem Cell Gene Editing Demo Kit. And what's really nice about this is that it includes our StemFlex media as well as our TrueCut protein and a positive control guide RNA so that you can optimize your delivery using the Neon system. And what I really like about this platform is that it has a very detailed protocol for electroporating Cas9 guide RNA complexes with the Neon system. It's got a really detailed protocol, very specific parameters for causing SNP changes in stem cells using this collection of validated tools. So this is a really nice resource that I recommend for all investigators that are looking to get started in stem cell editing. And another resource that we just recently published is an application note about TrueDesign Genome Editor and also has transfection protocols for stem cells. This talks through that G2019S LRRK2 mutation that I just showed you. So if you're looking to review those slides in greater detail in a PDF format, we also have that available now on thermofisher.com/TrueDesign. And it also has a secondary example using our TrueTag Donor DNA system. Our TrueTag Donor DNA system is a pre-designed donor template that allows for easy N or C-terminal tagging of proteins of interest. So what we're representing here in this cartoon is that we're going to be doing an N-terminal GFP tag into the ACTB gene. So we've got kind of an annotation of that. We're seeing where our start codon is for the active gene. And what we are providing here is a pre-designed donor that has a resistance marker, a P2A self-cleavage site, and then a reporter sequence, in this example will be GFP, that allows for a GFP fusion protein to be produced in your cell line once you've knocked this in. And what's really nice about this system is that we're able to drive very high editing efficiencies. We're able to demonstrate, greater than 80% and almost, in some examples up to 100% edited cells due to that resistance marker being present on the donor template. What this does is this allows us to successfully enrich for knocked-in cells and really overall improve the editing workflow of getting to an edited cell that has, your particular knock-in, that is this GFP fusion protein in it. So what you can see here is with or without Puro, we do have a decent knock-in rate. We're looking at around 15 to 20% depending on the example of these donors, and then we're able to drive that to a very high rate using that puromycin selection. And an example of this, we can see that we get really nice GFP actin fusions for this example. This is in U2LS cells, and you can see those really nice actin filaments forming due to these GFP fusions to ActB, demonstrating that we can get really nice fluorescent imagery, of these cells and being able to see at that resolution those actin filaments. So it's a really nice demonstration of this tool. But again, ultimately, we're able to drive that to almost 100% knocked-in cells using that puromycin selection tool. Now, to further refine this and where we're taking these tools and kind of a bit of a preview of where we're going for this is we really wanted to adapt this to be used in stem cell research. And we know that this is a really powerful tool for understanding various fate differentiation protocols and how we can take stem cells into, terminally differentiated cell types. So for this example, we've got an iPSC cell. We're going to go ahead and transfect that with our Cas9, our guide RNA, and our donor to make a TrueTag iPSC, differentiate them into neural stem cells and then into astrocytes. And what we needed to do for this was create a new type of donor that's going to have an exogenous promoter on that selection marker so that we can select for and produce these iPSC cells that are going to be, expressing this GFAP, GFP reporter, but do that in the stem cell state. Do our selection, our enrichment, get a really nice and enriched cell population before doing any differentiation. And the proof for that is that we were able to show that we were able to do our selection, enrich for these stem cells, differentiate them into astrocytes, and then when we do immunostaining for GFAP, we're able to see that this co-localizes very nicely with our endogenous GFP signal that we've knocked in here. This is one of our earliest experiments for this. We're currently further refining this design and these protocols here, and you can look forward to there being some new data on this front in 2021, but we've got some really promising early data here for this system that's going to be very stem cell specific for assisting in fate determination studies and developing protocols for differentiation. And for our last bit here, I wanted to talk briefly about how we're looking at this technology evolving and moving into the clinic. Thermo Fisher Scientific has had the cell therapy systems, or CTS, designation for products for cell therapy for about 20 years now. We've got our Gibco CTS brand that is designed specifically for investigators that are looking to move into the clinic. These are products that are 21 CFR Part 820 compliant. We have the testing and documentation to be able to support their use in the clinic and have the appropriate regulatory filing. And this has been used already in a number of FDA-approved CAR-T therapies as well as clinical trials. And we know that kind of the next area that we're going to see a lot of cell therapy development is obviously going to be in the stem cell field, looking to make additional CAR-T therapies as well as using stem cells for treating a number of different ailments and needing to engineer those stem cells appropriately so that they can be useful in a therapeutic setting. And so what we've done for that is that we are currently working on a Gibco CTS TrueCut Cas9 protein. This is something that we've now taken our RUO protein that was formerly built to ISO 13485 standards, and we've built this up to meet the European and United States pharmacopoeia standards for ancillary materials for cell and gene therapy. We now have this built up in larger pack sizes to meet what's needed for therapeutic amounts. We have the traceability documentation to support these, including drug master files and regulatory support files will be available. We have extensive safety testing, aseptic manufacturing, and sterile filling, all making this protein of the highest quality and suitable for cell therapy manufacturing use. Today, we have this available as an early access as our CTS prototype, and we plan on having a full CTS product available in 2021 with that large-scale manufacturing and being able to support clinical trial use and eventually and further manufacturing. For the CTS Cas9 protein, I just kind of wanted to give a quick preview of our release criteria for that. We have an incredibly extensive C of A for this. We're doing a full bioburden testing, full testing for residual DNases, host protein, plasmid DNA, full identity characterization by SDS page, and endotoxin testing less than 0.1 EUs per mg. And so we've got, again, a very clean protein here that's suitable for cell therapy manufacturing and something that we're really looking forward to and excited to be able to support investigators looking to move into the clinic with their gene editing platforms. In summary, we did a quick review of genome editing, and then we talked about our wild type and our new high-fidelity Cas9 enzyme that we're currently looking to bring to the market and to our customers. We talked about TrueDesign Genome Editor and showed how this can help investigators design new knock-in experiments, specifically SNP changes for stem cells. We showed some applications of that, the LRRK2 mutation with TrueDesign, as well as the example of where we're taking the TrueTag Donor DNA system and making it more stem cell friendly and help with those fate determination studies. And then lastly, we talked briefly about moving into the clinic and beyond with our CTS Cas9 enzyme that we will be having fully available in 2021. And with that, I would like to thank you all for your time. If you have any questions, please feel free to reach out to us, and enjoy the remaining presentations for the 5 days of stem cells. Thank you.